Very excited to talk to you about this particular presentation. This is an oral presentation at DDW 2025. My name is Dr. Mark Pimentel. This is a fantastic presentation by Dr. Germano and research from the MAST program looking at intestinal sulfate overproduction and what it does to the human body. There's a lot here. You have to come to the oral presentation to see the whole thing, but I'm going to try and give you a synopsis. When we're measuring three gases on Trio Smart breath testing, there's an interaction. So you have SIBO, you have ISO, intestinal sulfate overproduction, and you have intestinal methanogen overgrowth. And they all overlap in some ways. And so understanding the isolated ISO, the isolated EMO, and the various overlaps is critically important. But here we're studying what the human body's reaction is to these gases. So this is the study. This is 85 reimagined patients that have breath tests on the day of the upper endoscopy. They then get characterized for hydrogen sulfide and methane. You can't do hydrogen because you haven't given lactulose. So we determine if they have SIBO by culture using McConkey agar. So we have intestinal sulfate overproduction by hydrogen sulfide. We can determine if they have EMO by methane and SIBO by culture. And then biopsies are taken of the duodenum for transcriptomics. Now I'm going to jump to the real results for the sake of time. So what we see is intestinal sulfide overproduction changes the cells of the small intestine to the point where over 1,000 different pathways are affected. And so this is an incredible change compared to EMO alone or SIBO alone. So really, intestinal sulfate overproduction is a dramatic shift in how the bowel reacts. But if ISO is by itself, you get changes in glutathione, you get changes and reductions in endogenous hydrogen sulfide producing enzymes, and changes in fluid status of the cell based on aquaporin levels. If you have intestinal sulfate overproduction with or without with SIBO or with EMO, you have even more dramatic changes with mitochondrial pathways changed, effects on nociception. We're talking about substance P pathways, dysmotility regulation pathways, fluid status pathways, which could explain some of the diarrhea symptoms and decreased immune response. What the study then went on to do is to take the hydrogen sulfide producers we know occur in humans when H2S is positive, and then individually infect or colonize rats with desulfovibrio, Fusobacterium, E. coli K12, which is a hydrogen sulfide producer, and Proteus mirabilis, among others. And what we see here is that desulfovibrio and Fusobacterium creates all sorts of oxidative stress changes in the cells as a result of their colonization when we gavage the rats, and changes in transcription and changes in fluids homeostasis. Proteus mirabilis and E. coli K12 make even more dramatic changes in oxidative stress to the cells, as well as substance P dysregulation and degradation changes that could lead to hypersensitivity, for example, and again, a decreased immune response. But what's interesting is that many of the rat changes from adding these organisms are identical to the changes that we see in humans with hydrogen sulfide on their breath. So in conclusion, animals exposed to hydrogen sulfide producers reveal alterations in pathways and genes that are associated with the ISO condition in humans. And it does depend whether it's a brief exposure for rats or a chronic exposure as well. But what's important is we're starting to unravel the pathways that are associated with intestinal sulfide overproduction. And what we see here is that the most dramatic changes we see in the host response are seen when hydrogen sulfide is present. I thank you for your time and attention, and I look forward to sharing other good news at DDW with you.